So hi, my name's David Crean, Vice President of Corporate R&D for Mars Incorporated. I've worked in Mars in many different countries, in most of our businesses, over about 30 years.
Hi, Jeff Welser. I'm Vice President and Lab Director for IBM's Research Lab out in Almaden, San Jose, California. Been with IBM for a little over 20 years. We do everything from literally moving atoms around in our basement to big data and analytics. And we're recently moving into more industry solutions, including the food and ag space.
Hi, Bart Weimer from UC Davis in the Veterinary Medicine School. I've been in academia for nearly 20 years and have started businesses on my own as well. And my lab focuses on host microbe interactions and the microbiome. Just a little bit about what we're trying to do. We're trying to have this unusual collaboration to solve a common yet unusual, common problem with unusual approaches.
So I got to start because probably IBM is the one that's the most fish out of water here. We are doing a lot of work, as I said, in different industry areas. And really what we've been looking at over the past, say, 10 years, this whole phenomena or catchphrase of big data is everywhere. And of course, we all think it's going to bring great value. And we keep looking for that big value in different areas. And one of the things we look at is where's the data coming from? What's the new areas of data? So one of the areas that we've been investigating is genomics. And we have a large group that already works in what I call traditional genomics, if we can say that at this point, for personalized health, for seed and ag, sequencing individual species to understand how we can improve or deal with health issues. But the area of metagenomics came to light as one that would have even more data. And since we like data, that seemed like a good area for us to go into. So we started a symposium about 2 years ago called Sequence the City, which was bringing together experts from around the world to talk about what can we use metagenomics for? What would be areas that we could go and play with on this end. And we met up with UC Davis and Mars as part of that and said, well, one area that is clearly in need of new approaches is food safety. And it looks like metagenomics might, in fact, be an interesting way to go. So what we started then was a small consortium with the three of us that is growing rapidly. And it's all around the idea of looking and trying to characterize the microbiome using genomic sequencing at all stages in the food supply chain. And that's one of the reasons we see this as more of a consortium effort than something that's an individual partnership. So as you can see on the slide, we want to think about looking at what the microbiome looks like in the production side, the processing side, distribution, and even retail. So what we mean by that is we want to actually take samples of, say, raw food ingredients or swabs from the factory floor or the soil where the food's produced, characterize everything that's in there in terms of the microflora, bacteria, viruses, fungi, anything that has DNA and RNA, and just see what the community looks like, and begin to understand then how we can use that as a mechanism for monitoring the food supply chain at all points along the supply. So obviously, if we find bad guys, we would stop that from going forward, but we can do that today with regular testing. And what we really are looking for is instead the things that are unknown, or for early warning signals when there isn't a bad guy there yet, but something might indicate a bad guy is going to show up. Or maybe it's not even a bad pathogen, but maybe it's a heavy metal or something. So the idea is that right now you can only test for those things you know you're testing for. If you use the microbiome instead and just look at how that population may shift from day to day, that might tell you what's going on. There might be something new that's there and give you sort of an early warning flag. But what that means is sifting through a lot of data, particularly early on as we're starting to characterize this. There's huge amounts of data there. A lot of it's redundant. A lot of it is signals that don't really matter, and a lot of it's noise. So that's exactly the kind of problem that IBM likes to deal with. How do you deal with figuring out what's the important data and what's the not important data? And what we hope to do is actually take this then from both the science side into a tool that one could actually use at each of these stages within the food supply chain to monitor what the signal is telling you and give you the headlights you would need to be able to say, I've got a problem brewing, or maybe even I've got a way to make things healthier in the future. So that's the reason IBM got involved with this.
So I guess, just taking a step back, I mean, Mars has been making food products for the best part of 100 years. And you think by now, well, we've kind of got it sorted out. We know how to make safe food. And for the most part, we think we do. But actually, when we take a step back and look at the food supply chain, we're actually really worried. And what we see are pressures on the food supply chain like we've never seen before. Commercial pressures, social pressures, pressures of basically getting supply a lot of the time. And if you look at how we approach food safety, there's not been very many breakthrough implementations or new approaches. I think the last one was probably HACCP, the hazard analysis critical control point, which is great. But that was developed for the space program. It was developed for a space program just after chimpanzees were going into space. It was a long time ago. And what have we done since then to improve it? Well, actually not very much. And if you look at the test methods that we use, most of them are kind of cast-offs from clinical. And there's a bit of an attitude that actually food doesn't really need anything different. If we've got testing methodologies, we can apply those. We can apply them perhaps in ignorance of the statistical limitations, in ignorance of the detection limits, and in complete ignorance of everything else that's there, because when you're testing for one thing, you find one thing. You don't understand what else is there. And when you look at that, and then, okay, well, so how are we doing in society? Well, CDC produced some great data in 2011 that looked at the impact of foodborne illness. And in the US, 50 million people were hospitalized in 2000. Sorry, 50 million people were ill. The cost of that was about 80 billion. I'm not sure how many of those were hospitalized. I always get that mixed up. But 3,000 people died. 3,000 unnecessary deaths in the United States due to foodborne illness. Not all of that is because of the food supply, but a significant proportion is due to food supply. So that's in a developed country with great reporting systems. And that's really good data. If you go out into the rest of the world, what do we see? About 2 million people a year annually dying through contaminated food and water. And that's probably a huge underestimate. We see 25% of commodities, crops, contaminated with mycotoxins. That's an FAO number. 25% contaminated with mycotoxins. Is that 25% included in the amount of food that we produce? Because not all of that is thrown away. So 25% of the food that we think is available for consumption shouldn't be available for consumption. Actually, it's not safe, it's not food, so it shouldn't be there. So when we think about the challenges for the future of how do we feed the world, and was that lovely quote from Jason Clay of the World Wildlife Fund, that the next 40 years, the world has got to produce as much food as it has ever produced to feed the increasing population. And that population, it's not a case of if it's going to increase, it's there. The population is there today. The people are there today that are going to have kids. It will be 9 billion by 2050. So these are huge challenges. Let me just change that. And so if you put all of that together, and we think about, well, you know, actually we're finding it really difficult to keep our factories under control. We have to work really hard to make sure that we can make safe products. And if you take a step back, we have to work even harder to keep the food supply under control. I think it was the FAO who defined food security, that when all people at all times have access to sufficient, safe, nutritious food. And I think we've been working on the sufficient bit, and we've forgotten the other bits, particularly the safety. So the opportunity to work with these guys on safety was for us very appropriate, very timely. We think the food safety system needs a complete overhaul, and that's what we're setting about doing. And we think that genomics is the way forward. We think understanding fully what's going on in factories, the full microbial load of a factory, whether that microbiome in a factory is protective, whether it's not protective, whether it indicates risk, whether microbiomes can be protective, new indicators, new ways of identifying where raw materials come from. All of these things are very, very attractive to us. So that's why we got involved.
Okay, so where does academia fit into this consortium? And that is the fact that in genomics, while it's been around for a while, and some say we're in the post-genomics era, metagenomics has not been around that long, and analyzing the data at the scale that Jeff talked about, the microbiologists are not at all sufficient in doing. We need to partner with people that can do that. So that's one piece of where we need help in academia. But realizing that every single organism on this planet, including microbes, have a microbiome, and one way to look at that is to look at the entire community that's present within the plant or the animal. But also then, as Dave mentioned, if you look for just one organism, you'll typically find that and just interested in that. And that's where our current food safety system is. We're looking for one organism at a time, and we're not looking at the community because it's difficult. The technology is evolving, and it's there today to actually do that. Actually, the food testing that we use today was built in the 30s and 40s, evolved slightly in the 50s, and that's about where we are. And we've very much taken cast-offs of clinical testing and said, gee, we're going to use what's built for serum, and then we're going to look for it in all sorts of food products. And so food testing today is analyzed and approved, if you will, based on each food type. If we're going to make as much food as we need to in the next 40 years, we can't tolerate that sort of testing paradigm any longer. Next. So what we've really come along to is all things have a microbiome, all things in the, one more time. We have this slow classical testing that continues to miss outbreaks. And when we miss outbreaks, as Dave mentioned, we have lots of troubles. And so what tools do we have at hand today? We've got metagenomics, which measures DNA. DNA measures the potential of what the community is present, what the potential is of what those organisms can do in the food supply. We then have what this consortium is typically using is meta-RNA-seq, and I've been very careful not to call it meta-transcriptomics because we're not using it to look at how things are being done and why things are being done, but we're using it in a forensic way to understand what's being produced so we can look at the signals of what can be done within the food system. So we're looking for what's actually being done, what's being produced at the RNA level, and what's being transcribed then into RNA and proteins. And then there's a small portion of the consortium that's interested in looking at metabolomics, small molecules, heavy metals, bioactive compounds like short organic acids, for example, one more time. So why all of those are interesting and all of those are very technically challenging on their own, putting them into a context of use for food safety allows us then to do new method for sample processing needs to be developed. How can we get RNA and DNA out of every single food type that we use? That's a substantial challenge in the field today. In NGS, next generation sequencing, is continuing to evolve. And there's the major player in that area released new equipment in February, which changed the entire paradigm of what's being done again and what can be done. And then, of course, new analytics and new data types. One more time. So what are we doing in the consortium? We're producing innovative tools to redefine food safety. The challenge that we've taken on is very large, and we think with this unusual partnership that we can create actionable knowledge that the food safety system can actually use and get out of the 30s and 40s and 50s and into the 2000s very quickly. Oh, one more back. Okay, so what we would like to do now is open up the session for questions. And to start with, the tweet wall is very active, and you can put that up. And so to get things starting, we're going to ask a few questions of the panel ourselves and start really understanding what questions you have and start understanding what this really means for both the global perspective, but then also for the food supply chain. So maybe but for both of you, I guess. So this level of collaboration, it brings along a level of unprecedented transparency. And that hasn't always been the case for food safety. What does that mean? How is that going to impact society in this level of transparency as we move forward?
Why don't you start with that?
So, it's fascinating. Companies, food safety professionals were kind of consenting adults that did things very privately, locked in laboratories, reporting results, occasionally talking to regulators, but only when they really had to. And I think if you think about the data that we're seeing now, if you think about the interconnectivity of the world that we have, that we're living in, and the food supply, it's kind of got to change. So a better way to ensure food security would be able to have complete overview of all the data. And that actually, you look at it not from the point of view of making sure that you don't have issues, and if you have issues, you deal with them quickly so nobody knows about them, but actually making sure that you share that data so that people can see where the issues are, understand what the issues are, and you share that learning through the whole network. Because if you don't have that transparency, you don't share the learning, you're actually not getting better. And you're not preventing the network, you're not enabling the network to prevent those issues in the future. So I think this is a kind of pre-competitive space, is how we think about it. I think we're going to have to be much more transparent with the data that we present. We're going to have to find new ways of working with regulators, new ways of working with competitors, and new ways of working vertically in the supply chain.
I think too, actually, one of the biggest things we're trying to build up is a large database that will be a reference for everyone to use. So that gives us transparency across everybody from the supplier up through what you're seeing on the retail shelves. And that's a little scary at first for some people, and they think, well, I'm not sure I want my data as part of that. But of course, we don't have to tag the data with who it came from. But having that references out there, even in an anonymized form, gives everybody the resource. And I also think it gives them people, something else you get out of testing that you don't have in the past. Right now, when you do a test, if you didn't find salmonella, you throw that test away. That's good. It was good news, good day, move on. But you didn't learn anything, right? So this actually, every time you do a test, every time you do your microbiome test, you've learned something new. You've added to this database. So we definitely see that while we're looking right now from a safety aspect, we're hoping to find that, wait, this microbiome on this tomato makes it last longer on the shelves. Just naturally it lasts longer. Well, that's great. So now you can go back and start talking to the suppliers. Well, what were you doing that creates that microbiome in your field that I'm not seeing in this field? And that's not something that necessarily is a bad thing. It just means, no, maybe we can figure out how we can get that tomato over in that field also to have the same microbiome. So I think this level of transparency then comes with some advantages that we haven't seen in testing up till now.
Is this area actually only a first world problem, or is this a global problem that we need to think about?
So, I mean, we're a global business. We've got 133 factories in over 70 different countries. We try and take our raw materials locally. And what we see is that, we get lots of raw materials that simply we can't use. So we test them, they fail our testing, and we reject them. I'd love to say that that material is somehow disposed of hygienically and safely somewhere. It goes back to the supplier. I suspect it goes back into the food supply chain. I suspect that people that don't test or the look of the statistics test and don't find it, it goes back through the network. So this is a problem in the first world. I think it's a problem in the developing countries. I think we're finding out that it's more of a problem in developing countries. And the statistic one of the facts that I get really worried about is the recent link between alpha-toxin and stunting in children. Stunting is irreversible. You lose the potential of a human being, you lose the economic potential of that human, the social potential. It's a terrible thing. And it occurs at very significant levels. So in India, I think the level of stunting in children is just below 50%. In some parts of Africa, it's around 30%. And to think that this might be caused by a food-borne toxin is really disturbing.
So data computing is going to be a very key part of using genomics. Is data computing capable at a global perspective that we're thinking about today?
Yeah, so it's interesting. Right now, particularly because we're trying to do a lot of deep sequencing, we're really trying to build up data references. We're talking about terabytes, ultimately petabytes kinds of data, and the analytics it takes to run that is, of course, gigantic clusters of machines that really are similar to what we use for high-performance computing and physics simulations, weather simulations today. But the goal is that that's what we need right now to get the science and the background figured out. What are the baselines? What are the references? But one of the things that we're bringing into the consortium as well are testing companies who can think about, well, what would you actually do as a regular test? You aren't going to take a sample and do a deep sequencing every day. It's just not, even at the rate of the reduced cost of advanced sequencing, that's really not economically feasible, probably even in the foreseeable future. But if you can instead say, well, here are the markers you should be looking for. Here's the quick thing you can say, yeah, if these markers on these biocommunities are correct, you're probably fine, move on, versus, oh, wait, something's changed, now you need to go do a deeper sequence. So I think there's going to be an opportunity then with different parts of the food chain to be using it at different levels. And the other thing that's working, of course, in our favor is the fact that everything's moving towards cloud-based services now. So we don't envision people going to put HPC systems into their farms. They're going to use a system on the cloud that would be distributed out where they can send the data in, get the responses back, and just use it as a service, which is very economical, both for them as well as for us from the point of view of doing the analytics and storage as well.
Any questions? Yes.
Yeah. You asked the question whether it was a global or a first world problem. How are you going to, Mars, you're manufacturing processed foods for the most part. How are you going to get buy-in from small stakeholders or other producers? How are you going to get buy-in from the industry and from the small producers, local producers, even here in the U.S., into a system that's going to be maybe viewed as being imposed? That's I think the crucial issue here. Sort of like GMO technology and acceptance of that. How are you going to get that buy-in?
It's a great question, and I think we're a long way from imposing anything. I think right now we're at a discovery kind of research phase, and I think the invitation is there for as many people as possible to get involved in the consortium. Obviously, that's more difficult for smaller companies, but if we can find ways to start sharing this data to start sharing the publications, I think what it becomes is a stimulus to start to think about these kind of food safety issues differently. And I think it's a stimulus for innovation. And in this meeting, I would have thought that was quite an attractive thing, but all of these areas are a great opportunity for innovation. Once we've got a different way of looking at the world, and then we can have different ways of meeting what we find, those issues that we see.
Yeah, but no one's questioning your goals or whatever, but still, there's going to be perception.
Yep. Yep.
Can I add on to that? There was a question that came off the tweet wall that's very analogous to that, and I'll paraphrase it, is, are the regulators buying into this approach? And it's not only are the regulators buying into this approach, the FDA and the CDC announced this year that they're now regulating the food supply with next generation sequencing. So it's not only are they on board with it, they're doing it themselves. So if you're not doing next generation sequencing in a small farm or a large processor, the FDA is. And there's been a couple of cases already where there's been an outbreak. The company that was responsible was not doing next generation sequencing, and then they were caught out because the regulators had exactly more information than they did, like 44 million pieces of information compared to 27 pieces of information, literally. So the regulators are buying into that as well, and they're using it. Metagenomics is also on their plate. The consortium was invited to present at a food safety group meeting in Portland, Oregon this year because the regulators are using metagenomics and are struggling with it because it's a difficult thing to do. Okay, there was another question in the audience. I saw a hand. No? Okay. So with all of this great innovation, what are some of the key gaps of steps of getting to where we want to go from where we are today?
Well, I think from the IT side, I mean, we have obviously lots of computing power. We have lots of algorithms and analytics, but until we actually start to apply it in the scenarios, you don't really learn what's working, what's not working. Separating out what is signal from noise is something that's fairly common in pretty much all the things we do with data, but of course it's always very specific to the specific problem you're trying to solve. So from our viewpoint, getting lots of data in from different food sources, from different farms, from different locations, different times of year, we know all those things will cause the microbiome to vary. You need to look at all of that to understand these variations are normal, these variations are not. So it really is about getting as many samples as possible and as broad a spread as possible.
Absolutely.
Nothing to add.
Nothing to add.
Great.
It's hard.
It's hard. So you mentioned this idea of being pre-competitive in food safety. It's not been a common thing to think about pre-competitive in the food area. Can you describe a little bit more what you mean about being pre-competitive in food safety?
Yeah, sure. I mean, it's quite interesting, but over the years, I've seen a number of food safety issues, and there was a very well-known competitor of ours in the UK who sadly had a food safety issue, and about 50% of people thought that Mars owned this particular competitor. The other 50% of people thought that they owned us, and it was the sales of all products that suffered. And what was interesting is that, as you find out things that have gone wrong, Mars Incorporated has had a lot of experience over the years, and we've captured those experiences into standards and ways of working and people's experience, and trying to make those available to people, because actually nobody profits from a failure in food safety. And maintaining consumers' trust, and the consumers at the end of the day are the people that keep us here. In the case of Mars Incorporated, maintaining consumers' trust is really important. And so how can we do that? Well, we can do that by making sure there are less food safety incidents in industry and in the supply chain. But that's our very basic thinking.
Anything on the computing side?
Go ahead.
So can you give some example of the insight you're gaining from this research so far and how that can apply to food safety?
Let's talk about the science side.
So I think I can do. So the insights that we've seen so far, some of the questions we've been asking are, do specific supply chain products or commodities have a consistent microbiome that you could actually measure, follow, and then find a baseline? And we're looking at that right now. What we found so far is that there are microbiomes that are associated with specific inputs. The second question we've been asking intensely is, can we tweak the microbiome? Can we add things to it and force it to move in a certain direction? And the answer to that is yes, we can add. Especially, we've been focusing on adding pathogens into the microbiome. and then asking what does it do, how does it shift the microbiome, and can we use it in a predictive way? So if you look back at the things that are being done in the gut microbiome area, some of the similar questions are being asked, but we don't know which direction to push the microbiome yet. What should we add to make the microbiome to be protective against colonization with pathogens, for example, would be one key question. Can we establish a microbiome that would be resistant to letting microbes establish? And if you look at salmonella, that's a very realistic thing to do. Salmonella is not very infective. If you have a healthy microbiome and a good immune system, you typically don't get sick with salmonella, even if you consume a relatively high amount. And so we're taking a similar approach. If you have a healthy, whatever that word means, microbiome in an incoming stream into a process, then presumably you would resist some of the colonization by pathogens. And so we can see that. The second thing that we're being able to see is that for food authentication, this is a phenomenally technology for resolution. You can authenticate, did that fish that came into your product, is it really the right fish? Is it salmon, or is it cod, or is it just trash fish, so to speak? So it's very easy to see authenticity within the food. And so there's lots and lots of potential to look at this in a forensic way. So we are starting to bear out. There's challenges for sure. We're definitely seeing that there's analytical challenges that are not there on a single gene level. And so the analytical pieces and the data analysis pieces are very critical for that. Yes?
So how do you link your project with human health and all the work that you do in microbiology and human health?
Yeah, it's a great question, and I love the phrase from farm to fork, but probably we should be saying from farm to microbiome. We haven't made that link yet, but it's a great question, and I think inevitably that's where this work could go.
Absolutely. I think that actually one of the things we find would be really interesting is, can you figure out what are probiotics doing? Are probiotics really good, and what's the efficacy of them? Right now, we know they seem to work kind of, but no one really knows why and which ones actually do what. But I think if we could figure out how the microbiome of the food you're eating affects your own microbiome, that could be a really interesting whole new direction for it.
And do you know if the pharma sector is looking at the microbiome space as a pre-competitive space as well, or not?
I don't know.
Oh, sorry. The question was: is the pharma space also looking at the pharma industry looking at the microbiome as pre-competitive or not? I don't think anything's pre-competitive.
In pharma.
In pharma, perhaps. I don't know.
More questions? Yes.
Karen Sorber from Myconic Technologies. We have a water treatment device. I'm anxious to find out what you think about how the need for clean water affects food safety.
Yeah, I mean, sometimes it's very difficult to separate water quality and food quality. They are so intimately linked, and so the need for clean water is a fundamental, I would say, of food safety. Absolutely. And so I know there's a lot of different systems that are being looked at, but one of the basics of producing safe food is to have safe, all the ingredients safe. Often it's water that's one of the ingredients.
One of the comments I was going to make, we talk about the pre-competitive nature. I think one of the things, though, particularly for this audience, that I think will be interesting, so obviously we started with food safety, and as you heard, you kind of get food fraud for free along with that, because you're going to start to see exactly whether the food is what it says it is. And by the way, you also can decide if those olives are really from Italy or from California in the Italian olive oil you're using. But we think there's going to be a lot of other things you can get from this data. And we're going to try and figure out a lot of it. But I think as we get this data reference out there, I'm really hoping that the sort of innovation community out there starts to think about, well, what else could I do with this? What else could I learn from this? And I have no doubt there's going to be a lot of really interesting new applications of this in the future. So I hope that people here who maybe aren't involved with metagenomics today do start to look at this data when it starts to become more readily available and see what else can be done with it.
I want to ask one last question, and that is, what type of investor or what type of partner is ideal for the consortium?
So I would say right now we're trying to make sure that we get a few of the larger players in each of the segments. So we obviously have a big food producer with us already. We're talking to some of the larger testing companies. We'd like to get somebody on the producer side, in the farms themselves. Probably start to think about other types of food. Right now, we're looking at mostly raw materials that go into processed food, but obviously dairy is a big area that we'd like to actually be getting involved in, meats as well. So I think that trying to find partners in each of these so we get the baseline set is sort of the first step, and then from there we'd like to see it spread out to even smaller groups.
I think the key is that we need to be diverse.
Yeah.
Absolutely.
Very good. Thank you very much today, gentlemen. It's been a pleasure.
Thank you. Thanks.
Very, very much.
Thank you.